So thank you everybody for joining us this afternoon. I am thrilled that we have three molecular genetics / cellular biology scientists with us today for all of you to chat with. I think our goal in this session was to give you an opportunity to learn a little bit about what each of our scientists are doing, and also have the opportunity to ask some questions and have them answer them. Sorry, the light is in my face up there. So we're thrilled to have Larry Reiter from University of Tennessee Health Sciences Center, Rachel Weverick from the University of Alberta, and Rob Nichols from, is it Pittsburgh University? Children's Hospital of Pittsburgh. So I'm gonna, what we're gonna do is I'm gonna give them each a couple of minutes to introduce themselves and the kind of work that they work on, and then we're gonna take questions. And again, the idea here is for sort of it's fair game to ask almost anything about their opinions. What we hope to get at is what they see as the most interesting things in the field, where are the promising areas of research, and also if you have questions about the research that is going on right now. So I will let Rob lead off, and I have a couple of slides. I don't know if you want to use them or if you just want to talk.
The slides I sent were
just
for if we needed them for questions.
Okay, great.
Okay, so I'm Rob Nichols. I started work on Prader-Willi syndrome in 1987, so a long time ago. And at that point, there were no genes known. Imprinting wasn't known. I found that during my postdoctoral work, that genomic imprinting played a role in Prader-Willi syndrome. So I continued to work on PWS for my career, and started the first decade or so doing human genetics, and then switched for another 10 or 12 years to working on mouse models. We characterized a deletion mouse model of PWS that had failure to thrive, but severe failure to thrive as a consequence of hypoglycemia. The mice only lived a week. And so we were able to characterize the cause, and it was actually associated, the lethality was associated with pancreatic hormone defects, so defects in secretion of insulin and glucagon, and we're currently working on a cellular model. We've generated PWS pancreatic beta cells and looking at the mechanisms by which insulin is secreted. So that's one ongoing area that started from the mouse, but we're now using cell models for that. And then about a handful of years ago, decided that because our mouse models weren't developing hyperphagia obesity because they weren't surviving, that I wanted to generate a different animal model. And so I started working on trying to create a pig model, a mini pig. It's a Yucatan mini pig. And so first had to do the genomics, and then collaborated with a pig group in Missouri to establish a pig PWS model. And we've had the pigs in Pittsburgh since May, and now we're breeding. And so it's been quite an experience just diving in and starting to work with pig models. None of us had ever had any experience with pigs before. And they're like having dogs. I mean, they're like having, there's always things going on that we have to run to the animal facility to look after them.
So just for background, the pigs offer some advantages as far as the physiology is more like humans, and they have more complex behaviors.
Yeah, and it turns out that the brain development in pigs is much more like human, and the brain development and the brain size and the structure of the brain, the highly folded parts of the brain that are associated with intelligence, that's more developed, for example, in the pig than even lower primates. So lower primates like monkeys. So to get closer to human, you basically have to go to either chimpanzee or gorilla. And for a combination of ethical and cost and other reasons, it's difficult to work with great apes. So we hope that the pig is going to be an excellent model. And the disadvantage is that there's a much longer life cycle. It's a 3-month pregnancy, and then it's 7 months to maturity before you can breed. So it's basically a year per generation, and we don't have the ability to do genetics like with the mouse model. It's in terms of more complexity and greater cost. So the advantages, different systems, but we hope that the pig model, now we've developed pigs with the genetic mutation, and the pigs have the imprinting defect mutation, and so we're hoping that they provide a really powerful resource.
So Rachel Weverick, and I'm at the University of Alberta in Edmonton, Alberta, Canada. And I first became interested in Prader-Willi syndrome working in Uta Franca's lab at Stanford University as a postdoctoral fellow, and then moved to the University of Alberta in 1996, and have been looking at first at the genetics of Prader-Willi syndrome, identifying some of the genes that are within the deletion region, so the IPW gene, Nectin gene, MAGEL2 gene. You've probably heard some of these strange name genes coming through in some of the talks. And then we started looking at the effect of loss of those genes in mouse models. So we looked at mice that were missing the Nectin gene, that have deficiencies in muscle and brain formation, as well as in fat tissue. And then we looked at deficiencies in the MAGEL2 gene, and those mice have also deficiencies in the hypothalamus specifically, and in endocrine function. And after that, we started transitioning more to two programs of research, one more in the neurobiology of Nectin deficiency and MAGEL2 deficiency. And the idea behind that whole scheme is that when you have a deletion of the entire region, then it's very complicated to model, because there's so many genes that contribute to the disorder, looking at the disorder in humans, or the sort of complementary disorder in mice when you're looking at several genes at the same time. So the idea was that if you could break that down and look at just one gene at a time, and then add together the effects of all of those genes, then in some sense you could look at the whole by looking at the sum of the parts. And so we started looking more at the neurobiology of both the MAGEL2 and Nectin genes, and the proteins that are encoded by those genes, and then the proteins that those proteins associate with. So we have the MAGEL2 protein, and what we wanted to know was what are the other proteins that are near to the MAGEL2 protein in the cell. And by getting to know kind of those neighbors, we can get to know what processes the protein is involved in. And then you start to have some targets for how you can change those processes to benefit, first to benefit the mice, but then obviously to benefit people who are missing that MAGEL2 gene, or who have mutations in the MAGEL2 gene, as is the case in Schaff-Yang syndrome that some of you may be familiar with. So that neurobiology and molecular side is one avenue that we're still working on. And then the second avenue is looking much more directly at therapeutics that are either in clinical trials in Prader-Willi syndrome, or where companies have approached me and said, we're considering this line of research, we'd like to look at the melanocortin system. There's good evidence that the melanocortin system is disturbed in Prader-Willi syndrome and may be responsible for some of the appetite, excessive appetite that's seen, but we could really use extra evidence that there is actually a deficiency in the melanocortin system, and can we test our compound in your mouse model? So we've done that for setmelanotide, which was in development for use in a number of genetic disorders that include obesity. We looked at diazoxide as well, and you may have heard about the diazoxide trials that are now going into phase 3, or have results in phase 3, I guess, going into open in phase 3. And both setmelanotide and diazoxide had very beneficial effects in this mouse model of Prader-Willi syndrome, where the MAGEL2 gene is gone. So with diazoxide, we saw a reduction in fat mass that's elevated in those mice, And with setmelanotide, we saw a very large reduction in appetite, in acute food intake in the mice. And of course, the benefit of working in animals is that you can raise large cohorts that are all genetically identical, that can all be tested at the same time, and you can also look at the end of the study at what the consequences are to the animal and to the brain, and obviously that's not approachable in human subjects. So doing animal research is not unique to Prader-Willi syndrome. It's extensively used in the pharmaceutical industry and extensively used in research, and so that's really the rule that for most genetic disorders, there's animal research going on. And those mice were also used for a trial of a peripherally acting endocannabinoid receptor antagonist. So looking at the munchie system, but trying to get the anti-munchies by using the cannabinoid or cannabis system to address appetite. So there was a study from Yossi Tam using the mice that we developed, and this endocannabinoid receptor antagonist. And then we've also collaborated on a study in the same mice, the MAGEL2 mice, looking at the effect of vertical sleeve gastrectomy, which is a stomach reduction surgery, bariatric surgery, which also had a beneficial effect on fat mass and activity in the mice. We did not do the tiny surgeries on on tiny mice in my lab, but fortunately there's other people they are bigger than flies. That's true, they're bigger than flies. You do bariatric surgery on flies. Yeah, so those studies have been moving along as well. So that's our preclinical development side, and then the neurobiology and molecular cellular side to our program of research.
So I'm Larry Ryder, and I think I'm just going to tell you generally how I got into Prader-Willi research, because I've actually known Rob Nichols since I was a graduate student, and I started working on Angelman syndrome and 15q duplication syndrome as a postdoc, and so my lab was really kind of ramping up. We've been working on 15q for a very long time now, it seems like, and so we sort of knew the locus very well, and it really made a lot of logical sense for our next step to work on Prader-Willi syndrome. We sort of understood the genes in the region, and we have some systems in place. You might know me more as the tooth fairy guy, so I've been taking teeth and making stem cells. Well, there are stem cells in the teeth, so we make them into neurons in a dish, and then we can do lots of different studies. So we've collaborated with several investigators here, and we have our own research which is focused more on the autism aspects of Prader-Willi syndrome, UPD. You maybe have heard that there's a higher incidence of autism in kids with UPD, and so that was the scientific presentation I did yesterday. And basically what we're trying to do is collect as many individuals as possible, because when you do basic research, you know, one of the limiting factors in doing human genetics is that everybody's a little bit different, sometimes a lot different. So what you need is as many biological replicates as possible. It's very difficult to obtain those if you're going to make things called induced pluripotent stem cells that you might have heard of. So we took the approach of making these dental pulp stem cells because you guys can give us teeth. The teeth fall out, you put it in a tube and send it to the lab, and we grow the stem cells. It's simple enough that we have over 200 dental pulp stem cell lines in the lab from 13 different neurogenetic disorders. So we've been pretty successful with that approach thanks to the families, so it's really important that you guys continue to collect these teeth for us. We use them, we need them, and it really is essential to our research. Now the other side of my lab I'm just going to briefly mention is that we work in fruit fly models, not mouse models. And there are some serious advantages to that which we could discuss, but we don't currently have any Prader-Willi models in flies. Our models are focused on the seizures that occur in 15q duplication syndrome. And that's our current fly model that we've been using to actually screen for anti-seizure compounds. And so other than those two things, I really was hoping, to field questions, because I have publications and all kinds of stuff that you could look up online, so you don't really want to do that. You want to ask about science, right? All right.
All right, great, thank you. So, we'll have questions from the audience, but there were a few questions that were submitted through the app, and I thought it would be good to kick off with one of them, which is for each of you, what is one exciting finding in your area of research that's come out in the last year that you think might be interesting to people?
So you mean from our research or from others?
Well, could be. If you have some, if your research is the most exciting, which it often is, then that, or if there's something that is related but, potentially has application to PWS, if you have some ideas. Okay.
I go first.
You can go.
So, the most exciting thing I work on is what I just presented, right? Because it always has to be, right? And it is. So my concern was, from years of sort of being around Prader-Willi syndrome and working on autism on other syndromes, was that I never really saw anybody really looking for genes associated with ASD in Prader-Willi syndrome. So I'm very excited that we were able to finally do some RNA-seq, so look at gene expression in Prader-Willi UPD plus and minus ASD versus deletion versus control. And so that data we presented recently, and there's clearly a signature there. There's lots of genes that we found. There are also some known autism-associated genes in that set that we found. So this is very early. We got the data about a month ago. So we're not close to publication really, but we're excited that we actually found something, and it's unique to the plus autism group. So it may be the start of trying to figure out not only that mechanism of how those genes cause autism in Prader-Willi syndrome, but how they cause autism in general, and maybe in other cases of autism where that combination of gene expression is present. So that's, to me, is very exciting.
I think what I'm most excited about is seeing how many people see Prader-Willi syndrome as a tractable problem, because I think for many years the consensus seemed to be that this was an unusual disorder and nobody was ever going to understand it because it was completely unique and used pathways that were not related to any other pathways that anybody knew anything about, and so it was just going to be a really, really difficult problem. And I think that between the clinical people and the preclinical people and the molecular people and a lot of new scientists who are real experts in neurobiology who've been attracted to this problem, and they see it as completely tractable. They see it as, well, all we have to do is figure out the pathways and then just attack each of those pathways with a compound that has worked elsewhere and can work in this disorder. And so by breaking it down into its components, I think we're easily up to the challenge of addressing many of the pathways that are disrupted in Prader-Willi syndrome.
I can follow on from Rachel too, that it has been very exciting to see many new researchers come into the field, and also pharmaceutical companies that are willing to do drug trials, and just the new technologies that are available in science. We've had genome sequences that opens up not only studying of different humans, but different animal models that then allows us to utilize genetic approaches, the development of the so-called CRISPR-Cas9 genome editing. Now we can make any mutation pretty much that we want to develop new models, and we can also test therapeutic approaches using gene therapy type approaches. That's probably not the long-term way to go with Prader-Willi syndrome, but it opens up a lot of, increases our ability to understand what's going on. And then other technologies, as Larry mentioned, his dental pulp stem cells, induced pluripotent stem cells. This has opened up the ability to generate PWS neurons in culture, It's not exactly the same as in vivo in an individual or an animal model, but it presents ever better models. And by utilizing all these new approaches and new investigators, we're now really starting to get at the point, not just what each of the PWS genes do, because it's not one, it's a dozen genes. So it's infinitely more complex than a lot of the single gene disorders, which are still complex and difficult to develop therapies. But we're now getting at what these genes do, what their pathways are, as Rachel discussed, what they're interacting with. By understanding the pathways, the hope is now that we can develop, ideally, small molecules that will specifically target the defects, so that if they're specific targets, then they're not going to have a wide range of off-target effects or side effects, which you don't want. And so that's the exciting part now, having been in Prader-Willi research for so long, and it was just a matter of slogging away trying to even get a basic understanding. We can now see that we're going to understand what each of these genes are doing, and the systems evolved, and that opens up the real possibility of therapies.
So, and I'll just add to that, because I spent 20 years working in the area of gene therapy, and it was a long 20 years, but gene therapy as a field is really now starting to have an impact on patients and on many different genetic disorders. Now, that said, PWS is more complex. It's not a single gene where we put one gene back and it's sort of straightforward, but just the fact that all of that technology is advancing so rapidly. I was at the NIH for Rare Disease Day, and they've set a goal of using CRISPR technology to treat rare genetic diseases, hundreds of them. So they're trying to scale up over a 10-year period where they can have sort of an off-the-shelf CRISPR technology. Again, it's not perfect for PWS, because that is really, the technology as it is today is really adapted for one gene, one mutation that you can just fix. And we have several genes, and we still don't know exactly which one does what. But I think the overall point is that the technology is advancing, everything's being scaled up, and all of that, even though it may not be easily applied to PWS, will advance the field where we can start to tackle more complex things like PWS.
The other aspect, of course, is that gene therapy is easier when you have access to the relevant tissue. So for blood cells, it's quite relatively straightforward. Even for eye diseases, it's being used successfully. For muscle, other tissues, that it's not as great a risk as dealing with the brain. So I still, yes, there's possibilities in PWS, but I still think understanding the pathways and hopefully developing small molecules or other types of things, and you can consider a hormone like growth hormone or other hormones like we're working on now as small molecules, essentially. So that replacing what's missing or altering the function of pathways to develop therapies.
Right, I think it's good that we have both options. I mean, at the minimum, the gene therapy technology can be used to help you guys understand PWS so that you can,
In our animal models.
Yeah.
Very important.
All right.
I understand that gene therapy for the replacement is not there, but there's the maternal switch for the deletion patients. What do you think the future is there, and how can we support that research and actually get some animal models or even iPS cells that can look at the maternal switch problem?
All right, so do you know anything about Angelman Therapeutics? So they're using these things called ASOs now, and it's working very well, but as Rob said, you have to get it where it needs to be, and that's the key. So, the problem is actually getting it where it needs to be in the brain. So even for the ASOs, it's very difficult to get them into all the neurons that need to be in the brain, right? So theoretically, we could mess with the imprint, right? The switch that you're talking about, but we still have a delivery issue to get it there, right? To every neuron where it needs to be. And If you look at the Angelman field, which in some ways is a little more advanced in terms of, following when the syndrome develops in the mouse models, there's been a lot of work lately showing that when you rescue UBE3A, which is the gene that I work on a lot, it makes a difference. So it turns out, and a very recent paper shows, that you can rescue some of the brain function things where you can measure electrophysiology, you can measure brain activity. You can actually rescue that and not rescue the behavior in that mouse if you turn on UBE3A in an adult. But if you rescue UBE3A earlier in development, you can rescue the behavior. So that was surprising. I don't think people expected that. But those kinds of studies should certainly be done in Prader-Willi syndrome, right? So there's a lot of potential there, and it's a parallel track.
I understand that Ionis now has some ASOs that will cross.
I don't know.
So one of the issues, of course, is that the mouse brain is very small. It's 3,000 times smaller than the human brain. And so when you can apply ASOs and there's enough diffusion of the small molecules, they hit enough neurons, you can get an effect. And that's a lot harder with an organ that's 3,000 times bigger. So again, that's where an animal model like the pig may be important, because the pig brain is only 3 times smaller. So it's the same order of magnitude as the human brain. And so now you can, if you try your ASOs or other compounds, you can actually directly look at how many neurons you're now able to get to and what the effect is, and both in vivo in the animal model, and then as necessary post-mortem to look at the effects. And so,
I mean, I think the good news is the feasibility questions are pretty clear. Right. We have some good models now. We have the mouse, we have the pig, we have some molecules that will turn the genes on, we know, and we can start to look at these questions around when would it have to be done? To what level would the genes have to be activated to have an effect? So we're in a position at least to begin to ask those questions and look at whether this is a feasible approach for PWS, because that's what we want to do, right? So what's feasible, what's not feasible, sort through all of that and pick out the things that are the most promising.
I've got a big voice, I can speak. Okay, Rob, I was just wondering about the phases in the pig. Does it go through the failure to thrive, into the hyperphagia, into the obesity? Does it follow those phases? And also, just second to that, are all the SNO RNAs available in the pig, the same deletion?
okay, I'll answer the second part first, because that's the simplest. And the answer is yes. The whole region is conserved. The PWS region is conserved in all mammals, so even in mice, in rats, pigs. So the region, there's slight differences in different species, but all the major components are present. So then the first question, do we have all the same clinical progression? I can only partially answer that, because right now we have our founder animals, and we're in the process of initiating breeding to generate experimental cohorts, and so then we'll be able to answer the question much more definitively when we have a larger number of animals. So right now we have, it's kind of it sounds a little weird, but we have 1.5 pigs with Prader-Willi. and the reason we have a 0.5 is because, we have one pig that's full Prader-Willi, has a full genetic defect. And we have half because he's actually a mosaic. The way that you generate using genome editing these animals, he actually has 2 cell types. So half his cells have the full PWS genetic defect, and the other half of his cells are functionally normal, although they carry one of the mutations on the maternal chromosome, so that's silent now. And so both animals, as piglets, had hypoglycemia, which we not only see in the mouse models, but some of your children, maybe even when they're older, may have episodes of hypoglycemia. The full PWS pig had more severe and more frequent hypoglycemia, but we can treat that and get the blood sugar and energy levels back. And then about 13 weeks of age, they did develop what appeared to be hyperphagia, and so they then went on to a strict maintenance diet. At that point, they were still at Collaborators Laboratory in Missouri. We then have now, in May, shipped them to Pittsburgh, and our veterinarians wanted to start them off on just an ad lib diet without having the strict maintenance diet to start, and their weight just suddenly shot up within a few weeks. And I'm like, we got to get these guys onto a strict maintenance diet. So we can't say, we haven't characterized it in detail to really say it's true hyperphagia at this point. We need more animals so we can actually study the food intake and study the energy levels, study the body composition. But the signs are there that we have many of the features. I also point out that our full PWS pig has quite severe scoliosis, and a number of your children or young adults may have developed issues with scoliosis. So it looks like in the pig that's going to be a good model for the potential bone mineral defects and scoliosis.
Larry, I was just wondering how you're getting the autistic diagnosis for the UPD PWS population, because as I know you know, they're often misdiagnosed and overly diagnosed because of their social functioning. So how can you confirm that they are actually autistic and getting into the right population groups?
That's a good question. So we have been stuck with this problem from the start of using these dental pulp stem cells because we're remotely assessing people. However, there are better and better tools now. The SCQ is what we use. I don't know if you're familiar with what that is, but the SCQ works pretty well. It's not perfect. If you go to sort of any developmental disability talk where people are using tools to assess ASD, although the gold standard is ADOS-ADI, people use all kinds of other testing tools too. So you find that, if you ask the question, which tool to use, you get 5 answers, right? So we had to make a choice, and the choice was the SCQ based on the literature on how it's actually fairly similar in its ability to predict to the ADIR. So it's a much shorter questionnaire too. So we have a good rate of people filling it out appropriately and sending it back. So it's a valid point that we may have non-ASD kids in our ASD group. However, from the scores, they really split pretty well on that particular test. Now, that's all good and well, but once we did the molecular work, it was more striking because we seem to have picked correctly. If you look at the data, there is a striking difference between the plus and minus group, right? And yet the minus ASD UPD cases look like the deletion cases for those transcripts. So that means that what we're seeing is something unique to that group. That particular group is statistically significant and different from the other 3 groups that we analyzed, control, deletion, and minus ASD for these genes. And so that was encouraging. That said to us that we probably did something here. We picked something that's different between the two. I agree, it'd be great if we could do ADOS-ADI on everybody, but I don't know how many of you have had that done with your children, and it is not trivial to do the ADOS testing, and finding somebody who's qualified to do it even is not trivial. I know this because we've had a clinical study on duplication 15q for years, and it was probably the most difficult thing to do was to get someone to do formal autism diagnostics. So I would love to do that, but I don't think it's practical, especially in terms of trying to collect remotely. It makes it almost impossible. If you have any ideas, if there's some other test I don't know about, I'm happy to try something else, actually, but I think it's working.
Hi, hypothetically, if each of you had a couple of $1 million extra to spend on Prader-Willi research, where would you direct the funds?
Right. So, I mean, there are many expensive aspects of research, and particularly if you're including clinical trials in that umbrella.
Where do you think it should be directed?
I think that the model that FPWR has now, which is a basement to top floor kind of model where research or funds are allocated according to scientific merit and feasibility and potential to produce original results that are important in the end, is a very, very sound strategy. So, you know, across the board, I mean, that sounds like a cop-out. He's shrugging his shoulders. You're not really saying that.
I thought that was a great answer.
No, and I'm serious. I think across the board, everybody could use more funding, and more federal government funding in different countries is harder and harder to obtain, and getting seed money from FPWR makes a huge difference, and that's why some investigators who've gotten $100,000 from FPWR stay with Prader-Willi syndrome research, is because they got their first grant, and because of that first grant, they got an NIH grant, and then they'll keep working on it. So I think actually spreading it thin rather than thinking about one huge investment is actually a really good strategy. It's a well thought out strategy. It's not just a default strategy.
I can send you my address and you can send the check. I agree with what she's saying, and I've had interactions with other foundations of this size, and it is kind of what I would encourage. In fact, I think we were discussing, I encourage the Duke 15 Alliance to give fellowships to students, to graduate students, because that's 4 years of commitment on those projects and less money for them actually to commit per year. And it actually, in the long term, I think will have a bigger effect of people moving into that field, right? But at the same time, some things are expensive. So I'm kind of both ways. Like, I've really been trying to get to the point where we could do single cell sequencing in our cultures, but even there's nobody at my university even doing that, right? It's extremely prohibitive. So given that the structure is in place, if there is if there's a time where you had, say the standard projects, and then you gave away a project that had high merit that was expensive, I think there's enough good scientists in this particular field that you would probably be able to find one that would be worth spending a little more money on, right? Because it cost more, but it was, the only way to do it, right?
So I guess I'll put out another idea too, since I would agree completely with Rachel and Larry. I mean, if we each had a couple $1 million, we'd each do our work better. We'd have more people in our labs. We'd do it faster because we would have more people who can work harder. And we each believe in what we're working on, so we would progress that way. But I'm going to take your idea, and if we had a couple $1 million each, I've got to expand that and say, well, let's go real pie in this guy. And we had some, really wealthy donor come in and say, I've got $40 million or something like that for Prader-Willi. What would you do? What I would want to do is actually create a new institute where I handpicked the best Prader-Willi researchers and non-Prader-Willi researchers in different fields and bring them all together and put them in one institute to work together. And now, from these types of meetings like FPWR, we get to interact with other investigators and more people and share ideas and start collaborations. But if you're in the same place, you're doing that every single day. You're taking a lunch break, you're taking a coffee break, and you're talking ideas. So if you go real pie in the sky, that's what I would do.
I would support that and say that Howard Hughes has recently branched out into this. I don't know if you know about, HHMI investigators, Howard Hughes investigators. It's the Howard Hughes, right? This money was left for excellence in science. And the way it worked before, the strategy they had was they would pick these individuals who they would give enough money to that they didn't have to worry about the money, right? They've now moved in this structure to a place called Genelia Farms, and they've I think it's the first one, and I think they're planning another, and it's like what Rob is talking about. So it's a more open scientific environment for lots of people who work in slightly different areas to come together and work at a single place and excel research even further than if we all did it individually. So it's kind of an interesting idea that other people have started to do, or at least Howard Hughes has started to do it.
That's all great. We do try to do it, since we don't have $40 million. I mean, to the extent that we can, trying to bring all of you guys together and encourage collaboration. I mean, as a foundation, we do have different working groups. There's a preclinical animal network that Rachel's involved in. We just had a meeting of a Shaft-Yang advisory group. So we do try, to the extent that we can, without the $40 million, to be doing that and bringing the experts together and encouraging and putting funds towards collaborations. Jessica, did you?
I would say it might be an opportunity to refer people to the strategic, we've actually been preemptively planning what we would do, and then a lot of information is on our website.
Yeah, yes.
So,
yeah, we do, yeah.
So I should have read the strategic plan before coming here?
Yes, everybody, are you admitting you didn't?
To your point, the foundation has made plans of as large funds of money. We proactively, and a lot of information is out available for those of you that are interested in where we want to go. We put a lot of the brain power from all of our researchers to make that plan. So we just need the 40 million.
And maybe the family should hear that you guys have done a good job. So the researchers think, and I think, and I'm sure other researchers think, having just the fact that you have 2 announcements a year is incredible for a foundation, right? So you are funding good research, and you're doing it in a structured way that makes sense, and so you're spending the money the right way, you know?
If I understood what you collectively said correctly, it sounds like even though in the mouse models you use reduction of appetite as an outcome, that's not considered to necessarily be equivalent to looking at hyperphagia. So my question is, in your animal models, what criteria would you use to determine whether an animal was experiencing hyperphagia specifically?
So there are animal models that have hyperphagia. So there is a definition in a rodent of hyperphagia, which is excessive food consumption for activity levels, and they gain weight. So there are animals with leptin deficiency and leptin receptor deficiency and POMC deficiency and all the way up the appetite pathways that have what researchers in animals consider hyperphagia, which is eating too much. But if you're talking about hyperphagia as in thinking about food and asking about food and obsessing about food and asking why they, when am I going to get seconds, can't model that in an animal. And so that, it's a very good point that definition of hyperphagia is difficult to model in any nonverbal animal, which is everybody except us. But there are brain MRI type of imaging protocols that can assess hunger to some degree in animal models. To be honest, animals are just not a great model for many psychiatric and behavioral components of human disorders because of that exact problem. So we can model some aspects of hyperphagia, just the out-and-out food consumption and patterns of eating, but the thought processes behind that eating behavior are a challenge.
And I think that is an area, though, that we're going to be able to move into over the next 5 to 10 years with larger animal models like the pig and other large animal models, because the animals are more intelligent. You can do the imaging techniques for the brain are also improving all the time and are available, the MRI, functional imaging. And so although we can't ask an animal what it's thinking or feeling, there's a lot of aspects that will be able to be interpreted, particularly with the large animal models, I think, where the brain structures are more similar to human. So I think
even in rodents, they're working for food paradigms, they call them, how many times the animal is willing to press a lever in order to get a pellet of food. how often they go back. So there are some very sophisticated methods for assessing that.
And I just wanted to clarify that was not meant as a criticism of the animal models. It was more a curiosity of, for example, the advantages of the pig model versus the mouse model and what exactly that would be. But I think you helped answer that with the size of the brain and the intelligence of the animal and those types of things.
Yeah, I mean, in most of the PWS mouse models don't develop the degree of obesity that you might expect, given it doesn't mimic the PWS in humans in that respect.
Well, and anybody who's had a Labrador Retriever or Golden Retriever knows what hyperphagia looks like, because it's like 10 seconds, where did it go? Like, when's dinner? Like, you just had dinner, right? So it is possible to measure those things in animals, but rodents not. Yeah, it's not so easy.
Other questions?
A rather specific question. In the pig model, what's the nasal physiology of the pig? And in particular, the blood-brain barrier in the pig compared to the rodents?
Okay, you got me there. That's something I have to look at and perhaps talk with our veterinarians, so I don't have the answers for that, sorry.
Can we talk more about gene activation? So it was successful in a dish.
And in a mouse. Yes.
So what are the next steps?
Okay.
So it was successful in a mouse.
It was successful in a mouse. So Dr. Zhang, who's at Duke University, screened a library of compounds and found a compound that did activate the maternal allele. He used that, and it's all FPWR funded, yay! He used that in a mouse model of PWS, and the mouse model that he used is a model that has severe failure to thrive and normally dies. And what they saw was an improvement in survival, and the animals that survived also were, pretty normal size looking. So they didn't have that like really small phenotype or characteristics that you normally see. So that's the good news. So it seems to work to do the gene activation. It seems to have a therapeutic effect. The issue with that small molecule, and it is being advanced, so Levo Therapeutics is now working with them, so it's kind of gone beyond, when you start to get into big drug development, that's beyond our budget right now. So Levo Therapeutics is now entered into a agreement with them, and they're going to do preclinical development of that molecule. So they'll need to do safety testing and then try to move it ahead. The issue with the small molecule approach is that it's not specific for the PWS region. So it activates the PWS genes and it activates a bunch of other genes. So from a safety perspective, that's going to have to be carefully looked at, right? So what else is it activating, and is there any downside to that? So, we all want new genetic therapies for our kids, but actually our kids are, if we stay on top of their health, they're healthy, they can be pretty healthy kids. So what we don't want to be doing is giving them some exciting genetic therapy and, having some additional problems. So it works, and the safety issues will have to be really carefully looked at before we see if it'll go into humans. Then there are other, the study in a dish was from Mark Laland's lab, and that is it's a very specific approach. So it activates the PWS genes, and it doesn't activate other genes. But it is one of these, when we talk about delivery, it's a gene-based kind of therapy. So now you're getting into how are you going to get that gene into somebody's brain. So it's going to have delivery issues. So and there are other approaches using CRISPRs to do gene activation, and I think the Foundation's approach at this point is to try to help push everything forward and answer some of these feasibility questions. We don't know which of those therapies might actually be able to go into people, but at this point, for me, I don't think we can put all our bets on one approach or another. So we'll keep looking at the feasibility to see, A, if it will make a difference in kids after they're born, And then B, if it does, which are the best approaches? And then C, we'll work with whatever pharmaceutical companies we can to try to move as many forward as we can. So, you guys want to comment on?
Pretty good.
I would just add to that, you know, if you follow the parallel in the Angelman field, there was a drug that's it's called the Topoisomerase Inhibitor, and it worked really well at reactivating UBE3A, and it worked in the mouse model. It was great. It worked in a dish. It was really good, right? I think it worked in the mouse model. I know we did most of these experiments were in the dish. And the problem was, and is, in all these fields, for all these syndromes, off-target effects. So the same thing that we were just talking about killed that as a potential drug, right? Because, you know, you don't want to make things worse. So it's by saying that, I just want to point out that, you know, caution, when you get excited about a new compound, you really have to be cautious because you don't want to make things worse.
It's not a situation that's unique to Prader-Willi syndrome. I mean, in cancer therapy, this is absolutely classic, right? All the old drugs were great at stopping the cancer, but they also killed the patient because they just work on all cells. And so then as soon as people started developing monoclonal antibodies to attack really specific receptors that are only present on that particular type of cancer cell, and then the patient stays healthy and the tumor is gone. Like, that's an oversimplification, but we're aiming for that second line of therapy without having to go through people on the first line, right? Like, you don't want to do that first line, you just want those really specific targeted therapies.
And these models that we're talking about up here are really essential at figuring out which of these compounds might be the best, right? So that's going to be the breakthrough. As we build better models, you're going to be able to really look just at the activation at that locus better, right? And that's important.
Just on that, I was just wondering if we can tell anyway, if you alter the methylation of the maternal genes, if they're going to act the same way as the paternal genes. Do we have any idea of that?
Yeah, and the other complicating factor in this is if you turn on that maternal gene, that big transcript turns off UBE3A. So in theory, we could cure PWS and give the kids Angelman syndrome, which obviously would be a no. But the compound that I think was reported by the Duke group, it didn't change the methylation, it changed the histone proteins, and they did not see downregulation of UBE3A, so it didn't seem to be inducing Angelman syndrome. So, and I don't think that mechanism is all the way worked out, and you guys may know.
I think that was also true with Mark Lalonde's results. He was targeting a specific repressor, and I don't think that affected UBE3A. Right. The other aspect of the UBE3A story is, though, that although it's the sort of dogma in the field that the antisense transcript is what silences UBE3A, I think that's far from proven, and we're not in a scientific setting, but I could give you scientific reasons why I don't necessarily believe that is correct or proven. So I think that in terms of therapeutic approaches, that may or may not be a concern.
Yeah.
But if you change the methylation of the DNA or the histones and you reactivate, it should have the genes that are turned on should have the same effects as if they were expressed from the paternal chromosome, with that one concern is what happens with UBE3A. Would you affect that or not? But that would be the only concern there. And then the question is always, can you turn it on at the right amount? Because you don't want to turn it on to super physiological levels, for example, because that may have effects we can't predict. And so you want to turn it on just enough to replace the gene functions, but not super.
Right, and I guess the encouraging thing from the Duke paper, which wasn't a mouse model, was it did seem to have a positive effect, and the animals, you know, were healthier after the treatment than before. So it was a step forward. Other questions?
Just a stupid question. Why does the body suppress the mother side dish? Does anybody?
What the whole imprinting? Yeah, but why do we have imprinting? That's a.
I think didn't Art Bodette write some thesis on this? Did you see that?
Yeah.
There is a review article that Art Bodette wrote about his theory as to why this is. But there's a lot of theories.
Yeah.
Point of view, I'm just curious. I just want to understand why body does like one side. There's two sides of it, and just decides not to use one side of that.
And it's not the only imprinted locus, right?
There's about what?
There are a number of
200 genes.
theories. Some of the best theories, I think, are from a guy named David Haig, who's an Australian who works at Harvard. He's an evolutionary biologist. And so imprinting occurs in mammals. It also occurs in plants, interestingly enough, and has the same molecular mechanisms, but the way it evolved is somewhat different. The idea in mammals is that paternally expressed genes evolve to make their offspring bigger and stronger and better able to survive, whereas maternal genes evolve to suppress that. So this is associated with genes that are expressed in the embryo and in the placenta, and maternal genes evolved to suppress that so that every offspring had an equal chance at survival. And the theory relates to not humans, but other mammals where they are not monogamous, and so there'll be many partners. So each male wanted his offspring to succeed. The female wanted all her offspring to have an equal success. So there's this kind of arms race between paternally expressed genes and maternally expressed genes. And so this is kind of a general theory, and it holds. It turns out a lot of imprinted genes are growth factors. A lot of paternally expressed genes improve embryo growth, and a lot of maternal genes help suppress that. With something like Prader-Willi, it's not quite so clear because most of what's going on with Prader-Willi is postnatal behavior, but there are other imprinted genes that affect maternal behavior postnatally and offspring. So it gets very complicated in this whole theoretical evolutionary arguments. That kind of tried to give you a quick idea of some of the ideas that are in the scientific field.
And the growth hormone fits perfectly veterinary, right? It's like dad's.
Yeah, it's mom versus dad. Really, is the theory. It's interesting. So I believe it's time for us to wrap up, and so I want to thank our panelists for sharing their.